Transcription. For the exam, you should know generally how transcription works, but you don't really need to memorize anything. Most of the tests will be testing your practical knowledge of it, and we have example problems coming up. Where with our replication, the purpose of replication was to duplicate the entire genome indiscriminately. Transcription is a lot more specific. You do not want to transcribe the entire genome into RNA. That'd be kind of pointless. You want to only transcribe part of DNA. So you go from some of our DNA, or an organism's DNA, to RNA. Yeah, but RNA has a lot of different functions. It can be mRNA, which is meant to produce our proteins. tRNA, transfer RNA, is used in translation, or perhaps ribosomal RNA, which is RNA used for our ribosomes. So RNA has specific purposes, and that is why we only want to transcribe some of the DNA. We only want to pull a little bit of the DNA out for it to do its function. So there are 3 general steps that we want you to know about. The first one is initiation. Right. So what synthesizes new nucleic acids? We talked about this a couple of times, but that would be a polymerase. There are multiple RNA polymerases involved, but we're not learning that. So just an RNA polymerase, because we're dealing with RNA and we're making more RNA. And then a polymerase, or when you synthesize a nucleic acid, you got to follow 2 rules. One, it has to be synthesized in the 5' to 3' direction. You don't bring that memory device that would you rather work a 5 to 3 job or a 3 to 5 job. And then 2, it has to, the parallel strand that you are producing, like a new one, has to be anti to the template strand. Okay, so earlier I said that you only want to transcribe some of the DNA. So it is a specific segment. And what indicates what section we want to transcribe is a section of the DNA called the promoter. So this is called a promoter. And it can be different sequences of bases, but the one that you'll hear very often, and this is an oversimplification, but it would be a Tata box, where T would be thymine and A would be adenine. So this section might be composed of a bunch of alternating T's and A's. And that indicates that we want to focus our transcription efforts around our promoter. So there are 2 strands involved. Our RNA polymerase is capable of breaking these H bonds and separating the DNA strands. However, this strand over here does not have the promoter. It is not what we use to make our new nucleic acid. And we call this one the coding strand. We'll talk about why in a little bit. Now this one is a template strand because we are actually using it as a template to synthesize our new nucleic acid, which would be the RNA. So template strand, you're actually using. Coding strand was just attached to the template strand before. So looking at our rules over here, we know that based on the promoter being on this thing, and also initiating with follow promoter. Based on our promoter being on this strand, we know that we want to focus our synthesis of a new RNA on this strand, and we got to figure out what direction it goes. Does it go this way or this way? And we know that it has to be anti, and it goes in the 5' to 3' direction. Looking at this strand, although it loops around a little bit, this is the 3', the 5' strand. So that means that we need to synthesize it in the anti-direction. So this is the 5' end, so we need it to be anti-parallel, so we would go this way, because we have to do it in the 5' and 3' as well. So the promoter is not actually transcribed. It only indicates where our polymerase will bind to. So initiation would also involve our RNA polymerase. So our promoter, it indicates that our polymerase should start transcribing around this area, and it'll go in front of it a little bit. It does not actually transcribe the DNA that the promoter holds. So then our RNA polymerase will bind. And next we have elongation, which is what it sounds like, where our RNA polymerase will go down the line, and as it goes, it'll synthesize our new nucleic acids according to our template. And we'll zoom in on this in a little bit, but our template strand has different bases on it, and then they are paired with nucleotides that are complementary to them. So yeah, and eventually it'll reach a sequence called a stop oh no, not a stop sequence, a termination sequence. So the third step is termination. During elongation, our RNA polymerase is going down, is synthesizing new nucleic acids or RNA, and it would be going in the 5' to 3' direction, and eventually it would transcribe a portion of the DNA that is that allows it to break off. So the RNA, the mRNA, or like the RNA that you produce, will break off of our template strand, and that is the end of it. It is only this long. We only transcribe this section of DNA. What allowed us to specify this little section of DNA to transcribe was the promoter, which kind of acts as like one like cap, and then our termination sequence, which acts as the other cap. So to make this little truncated RNA. Let's take a closer look at what's happening. So let's say that this is our template strand. We know it's a template strand because it is indicated by the presence of our promoter, oftentimes a TATA box, and the promoter reveals or indicates that this is our general region of DNA that we want to focus on. But do we want to transcribe going this way or this way? And we got to keep two things in mind. Nucleic acids can only be synthesized in the 5' to 3' direction, and the synthesized strand must be anti-parallel to the template strand, meaning that the 5's can't be on the same side and the 3's can't be on the same side. It has to be 5 and 3 and then 3 and 5. So by the chemistry, we know, let's say we try to go this way. We know that we have to go 5' to 3', but if we did that, our 3' directionalities would line up, and that is not anti. So we have to go this way, 5' to 3', and we see that 5, 3, 3 with 5, that is anti to each other. So our RNA polymerase will bind to the general area of the promoter, but it will not transcribe the DNA that constitutes the promoter. It'll transcribe this region over here, and it'll keep on going. This is our elongation step. And it'll keep on going until it reaches a point called the termination sequence, and it'll also transcribe this. Compared to the promoter, the termination sequence is transcribed, and due to the chemistry of like the bases that you get out of it, it allows the RNA to detach, which ends the transcription of that particular RNA. Let's take a closer look. Here we see our template strand of DNA. We see an RNA polymerase, and we see a bunch of RNA nucleotides that the polymerase will use to synthesize the RNA strand. The RNA polymerase detects the promoter, and then it finds out what region it should be transcribing. It reads the first base would be an adenine, and it looks for the nucleotide that would complementary base pair of it, and that would be a uracil. So the hydrogen bonds form between the adenine and the uracil, and then after each complementary base pair, then the RNA polymerase will actually make the phosphodiester bond between them. So the RNA polymerase goes down one nucleotide at a time, reading the template nucleotide, and then finding an RNA nucleotide to match up with it. Eventually, the RNA polymerase will synthesize the termination sequence, and the termination sequence will allow the RNA to break off, ending our RNA transcript. Let's talk about the implications of one strand being called a template and the other one being called the coding. Let's say that this is our segment of DNA that we are transcribing. This strand has our promoter, so it is our template strand. After we break these H bonds between them, this is an actual strand of DNA that will directly be used to synthesize our RNA. So let's model that. This is our template strand. A polymerase can only synthesize DNA in the 5' to 3' direction, and a complementary strand, nucleic acid strand, must be anti-parallel. So this is the 3' end, then we start at the 5' end. What nucleotide would go with A, or is complementary to A? Because this is the RNA we're talking about, it would not be thymine, it would be uracil. And then cytosine, that would be guanine, uracil, cytosine, and then adenine is just fine. So our RNA was synthesized in the anti-parallel fashion and in the 5' to 3' direction. So the only strand of DNA that was directly involved was the template strand. But the coding strand is very useful for our problems and whatnot. Because, okay, let's look at the coding strand. So 5 prime, that would be T, G, T, C, A. And then our RNA would be U, G, U, C, A. Okay, what do you notice about this? It would be that our RNA is identical to the sequence of the coding strand, except instead of thymine, it has uracil. So this makes sense because our template strand of DNA was complementarily paired to the coding strand, and then when you were synthesizing new RNA, you were complementarily pairing it with the template strand. So the coding strand and RNA have the same sequence, except RNA has uracil instead of thymine. So what are the implications of this? That means that if you're given the coding strand, let's say that you're given in the reverse order, you're given that it is T, T, A, U, oh, sorry, that is not a U. Let's call that G. This is a DNA coding strand, and then T. So if this is the coding strand, that means that it is identical, that the basis would be identical to the basis of our mRNA made to the template that was attached to the coding strand. So it would simply just be replacing the T's with U's. However, if we were told that this was in fact a template strand, we would need to do this process. We would need to pair it up in actually a complementary base pair, keeping in mind that this is RNA and putting U instead of T. So a lot of times, to keep things simple, you're given something like, okay, this is a coding strand of the DNA section as transcribed. What is the amino acid structure produced from it? You wouldn't have to take the additional step of transcribing it per se, because all you have to do is replace the T's with the U's. But if you were given a template one, you would.